COVID-19 disrupted our world, and in many ways it still does. But one key tool that helped us manage the pandemic was the PCR test, or the polymerase chain reaction test. What you might not know is the PCR tests aren't just used for detecting COVID-19. They're also used in genetic testing and even mapping out entire genomes. But how exactly do they work? Let's break it down. First, we need to understand a little bit about DNA replication. In our cells, DNA has to be copied when cells divide. This happens in a process called the cell cycle. During the S or synthesis phase of this cycle, DNA is untwisted, unzipped, and separated, and each strand is copied to create two identical strands. This process involves enzymes like DNA polymerase, which help build the new strands by adding the correct building blocks, also called nucleotides. Now, in PCR, we're not interested in copying an entire DNA strand, only focusing on specific parts of the DNA. In the case of COVID, that might be the location that's most likely to contain the virus's genetic material. So instead of replicating everything, we only replicate a certain segment. Here's how it works. The process starts by heating up the sample so the DNA strands separate. This is called denaturing. Then the sample is cooled down so primers, or short pieces of DNA, can attach to the target region and communicate and act as a signal to the Taq polymerase, which is basically a special kind of polymerase that can withstand the high heat used within the PCR process. And this starts building a new strand of DNA in the stage called extension. At the end of the first cycle, there are two identical copies of the original DNA. But the magic of the PCR test is that you can do this as many times as you need to in order to obtain even billions of copies of DNA, and this can happen in just a few hours. So let's say we use PCR for COVID-19. In this case, we're looking to see if a specific part of the virus's genetic material is represented in the sample. If the sample does contain this viral DNA, it will match the primers, and the test will show that the person is infected. But as I mentioned, PCR isn't just for detecting viruses. It's also used in areas like forensics, where it can help match DNA from a crime scene to a suspect, and you only need to look at a specific sample in order to match one DNA strand to another. It's also used in genetic research, where scientists can study DNA to learn more about ancestry and also look for specific genes within someone's genome, like whether they have lactose persistence or if they have any certain diseases. So ultimately, a PCR test works by copying a specific section of DNA and amplifying it so that people and scientists can tell whether a virus or other genetic material is present. And while it's an incredibly powerful tool used in science, we can also see it be used in medicine and criminal justice.